Man,
Hello, everybody.
John Paulting in the house.
Hello.
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 Hola. I hear a Yamaha.
Okay, let's see.
Oh, great.
Very good. Okay, let's see. We've got.
It's fun seeing all your faces.
Yeah.
It's
David,
been,
did you want me to
very
unmute?
I'm. Can one just automatically unmute everybody? Or sorry,
yeah.
mute everybody. Mute everybody.
Yeah, it said you wanted everybody to unmute.
Oh, sorry. I chose the wrong one. Sorry. Sorry. How do I ask everybody to mute? Can we mute everybody easily?
I think so.
You can ask us.
Oh, there you go. Okay. Okay. I'll mute everybody. Okay. And Antonio, if you could unmute.
Yes, I can do that.
Okay. Very good. Okay. Welcome everybody to the PhD defense of Tonio Gomez. I have a few slides to show to begin, and before Tonio takes over. So Tonio arrived in my lab with a tremendous amount of entomological experience. Just wait, here we go. Okay. He did his undergrad degree, his bachelor's at the University of New Mexico, where he worked with Kelly Miller, and from that produced several publications.
David?
Yes?
Sorry to interrupt, but you're sharing the wrong screen.
I'm sharing the wrong screen. Thank you, Virginia. I am sharing. Is this better? Yes? Is this better?
It's right to me.
Yes? Okay. Okay. So Tonio began his formal training at the University of New Mexico with Kelly Miller. He produced several publications there, including a couple on water beetles. He then did let's see if I've got this right. I think he then did an internship with David Kavanaugh at the California Academy of Sciences, where he worked on some little ground beetles, and then went to the University of Arizona, where he did his master's degree with Wendy Moore. Here's Wendy here on a group of ground beetles, this group right here. In 2014, he came to Oregon State University, where he joined my lab. His journey here at Oregon State University began in terms of research by studying a group of ground beetles called the Leibionines. And these are a gorgeous group and a very interesting group of ground beetles that are very compelling and worthy of study. But for Tonio, they didn't quite catch on. And it was clear that it felt like a bit of an academic exercise to him. But through time, he found a passion. He harked back to some things that he had encountered when he was an undergrad, and he became fascinated by the sperm of ground beetles and the reproductive tracts of the females. And this is one of those moments, as I saw Tonio gain this passion, that is one of the most pleasing things about being a mentor at a university, is when you see this spark come into someone's eyes. And it was this passion that was deeply important for Tonio for the next few years. He was especially interested in this little group of beetles. And in order to study the sperm and the female reproductive tracts of these beetles and other ground beetles, he did a number of expeditions, including to South Africa. Here he is in South Africa. Here he is in the car with his colleagues in South Africa. And this picture also points out something that's been very important in Tonio's journey, and that is the people around him. So here he is with a group of integrative biology undergrads in an open house, and this illustrates something else. Tonio is passionate not just about the beetles, not just about their sperm, but about other people and about spreading his knowledge of and his wonder at the organismal world. Here he is at the open house, very excitedly telling some kids about insects. And I think Tonio's role as a mentor was incredibly important here, and perhaps most importantly in the context of my lab was his mentorship of Kendra, shown on the right here. So during Tonio's time, he here at Oregon State University, he went through the traumatic last year and emerged similar to a standard hero's journey, much better than before. And I am so pleased he's gotten to this stage of presenting to you all of his interesting work and defending his dissertation. And so with that, I will pass the baton to Tonio. Take it away. Oh. We've lost you, Tonio.
Started.
Oh.
Oh no.
You're back. You're back.
That does not bode well. All right. Well, here we go. Thank you, David. I'm happy to be here talking to you all about an aspect of biology that I think is particularly interesting, which is the diversity and evolution of sexual traits. And I focused on, as David mentioned, ground beetles. I think they're wonderful. But we could think of other sexual traits out there. I gotta admit somebody. There we go. There we go. But before I begin, I want to take this moment to acknowledge that a majority of the research that I will be presenting today was conducted on Oregon State University campus, which is located within the traditional homelands of the Marys River or Ampinefu Band of the Kalapuya. Following the Willamette Valley Treaty of 1855, Kalapuya people were forcibly removed to reservations in Western Oregon. Today, living descendants of these people are part of the Confederated Tribes of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. So thank you. I also feel like it's necessary to say there's going to be some talk about sperm, a little bit about penises, not so much beetle vaginas, but definitely beetle sperm storage organs. A friend of mine asked me that recently, do beetles have vaginas? And some do, so there you go. As an outline for the talk, I'm going to give a brief introduction. Then I'm going to dive into my focus of sperm evolution across ground beetles, this major lineage of terrestrial insects. And then dive in a little deeper into a clade within ground beetles, where I focused on male trait and female preference co-diversification. All right, so what's the deal with sexual traits? Well, we know that they're odd. They show a lot of just spectacular diversity from things like song and dance. But let's also not forget sperm penises and sperm storage organs. These are some tremendously interesting sexual traits. And I think it's, and you know, when you do a Google search, this confirms it, although actually wasn't that interesting. But I can all agree the world would be a much duller place were it not for sexual traits. The importance of sexual selection here is that it's presumed that these traits are evolving under some sort of selection for an optimal trait, and that's evolving with a female preference. And there's this notion that perhaps species that are experiencing higher intensity sexual selection have more species, and the converse might be true. That's still very much open to speculation. But I thought it might be helpful to start with a very brief crash course on sexual traits. So we can begin with Darwin. I feel like that's a necessary starting point. Not so much actually his publication in 1859 that's very well known, Origin, but more so with his later work in 1871, The Descent of Man. Now, this was a very important work because he attempted to resolve some of these traits that he kind of struggled with. And they happen to be sexual traits, that they seem to be too gaudy, too ostentatious to evolve under the mechanism he focused on in Origin, natural selection. And so he developed this idea of sexual selection. And with it, early on, came the notion that there's a male trait or ornament, and that's evolving with a female preference. And then as we move on in time, came some of the first development of evolutionary models in the 1920s and 30s to attempt to explain how these two traits co-evolve. And some of the simplest models predict that there's a gene for a trait and that there's a gene for a female preference, and that the two become evolutionary correlated over time. The world kind of stayed a simple place in terms of sexual traits until the 1970s. Saw Jeff Parker observing the yellow scat dung fly doing what it does best, which is mating a lot. And he had the realization that if a female is mating with multiple males in a single reproductive cycle, then it ain't over yet, that there's still competition after mating, which was huge. And Darwin and many workers of his time were very focused on traits involved in pre-copulatory events, but there wasn't much focus on post-copulatory events. And as we continue further into the 70s and 80s, we see some development of continued refinement of evolutionary models and consideration of other scenarios that might also produce exaggerated male traits. And some of you might be wondering, well, this is strange. We're talking about male traits. We're talking about female preference. Where does that come in? It's really not until the 80s that we begin to consider the female perspective. And importantly, it was with, it somewhat started, you might say, or at least maybe a trigger was Bill Eberhard's book, Female Control. It maybe is lacking in terms of concrete examples that definitively back up post-copulatory sexual selection as the mechanism shaping these traits, but it sort of forced us to think that the female isn't just a passive vessel sort of witnessing these events, which is what a lot of biologists thought. And now, in the past decade or so, we've seen some incredible strides in terms of sperm female reproductive tract coevolution, including comparative studies that use many species and multivariate traits, not just one trait. Some incredible 3D interactions. This is a micro CT of a male Drosophila on the left, and he's in copula with a female Drosophila on the right. And there's all this spectacular data we have now on how the Aedeagus, the penis, interacts with the female reproductive tract and all sorts of like awesome stuff. One of my favorite examples is we have information on in vivo sperm interactions. Most sperm have been studied in vitro, but really there's some key events that happen as a result of interactions between sperm and the female reproductive tract epithelium. And most of those have been just a complete mystery until we started manipulating fly genomes and creating GMO flies with glow-in-the-dark sperm. Very cool stuff. So what we've come to find, if I can sort of frame where we're at right now, is that post-copulatory sexual selection involves male-male sperm competition and involves an aspect of female choice, but it's not like it's one or the other. And they're not mutually exclusive, and oftentimes the two play off one another. So as I mentioned earlier, we know a lot about pre-copulatory traits, not so much about post-copulatory traits. But what's clear is that there's a ton of really fascinating morphological variation with potentially huge consequences in terms of being a putative driver of the genesis of new species. Now, when we look at sexual traits like sperm, the diversity abounds. There's all sorts of wacky examples here including sperm without flagella, which seemed to be sort of a critical ingredient for sperm, giant 6-centimeter sperm fly a few millimeters, as well as all sorts of odd modifications to the head we can see in this example. So just awesome stuff. I love looking at this. They also do something else odd too, which is that sperm sometimes don't travel alone. Sometimes they travel in groups. Here's some examples from different groups of organisms, guinea pigs, rats, and beetles. And, you know, this is interesting because they start off as a group, but then they have a solo career, basically. And all of this is happening in the female's reproductive tract. So presumably, these traits are being acted upon by the female's reproductive tract. There we go. Yes. So where theory is at for a lot of these traits is that the female reproductive tract is the arena in which sperm compete. And some of the earliest observations that matched this theory were the observation that external versus internal fertilizers tend to have differently shaped sperm. And in general, internal fertilizers tend to have much, much more morphologically diverse sperm. And we might think that that may have to do with the fact that these sperm are going on a very strange journey compared with organisms like frogs that simply send their sperm out into the water to go fertilize eggs, versus maybe sperm in a butterfly that has to be stored, potentially might get dumped. It has to navigate to a certain site, perhaps. Maybe it needs to be there within a certain time window. So lots and lots of reasons for why you might think internal fertilizers have more complex sperm. So what's needed now that we're at this point? Well, we need more sperm female reproductive tract studies, and in particular, the female reproductive tract bit. We know sperm from thousands and thousands of animals, but the female reproductive tracts of most species haven't been studied. It's also clear that in vivo interactions between sperm and female reproductive tracts are really critical. And so we need more studies that are doing that. And we need more studies that are focused on actually attempting to estimate the evolutionary history of these traits. Even though ornaments have attracted tons of attention from biologists, there's actually surprisingly few studies that are focused on reconstructing their diversification. So I attempted to go after some of this for my PhD. I didn't succeed in every case. And in general, my goals were to advance sperm morphology in ground beetles and reconstruct sperm female reproductive tract co-diversification across this group. So I'm going to switch now from my intro to focusing more on a particular study where I looked at sperm diversification across a lineage of terrestrial insects. And so these are the ground beetles, and here's one example. There's actually 40,000 species, and so it's difficult to summarize that in a single slide. But they're very interesting. In particular, they have some of those unusual sperm that I mentioned earlier, where they're packed in a group. They also have a lot of species, which is great when you're dealing with statistics and you want a large sample size. They're widespread and ecologically dominant in terrestrial ecosystems. And yes, my favorite, they have really unusual genitalia, some funky sperm, and externally, I think they're interesting too. So the goals of the study were to advance chirality as a system for studying sexual traits, to document sperm morphological diversity, and to reconstruct sperm diversification. As part of this work, I needed to review the literature and figure out what was already known about sperm. And the earliest record I can find is actually from 1884, when Gilson decided to look at some arthropod sperm. And I remember looking at this early on. This is like, and I'm just barely getting like nerding out into the beetle sperm stuff. And I remember seeing this illustration and thinking, gosh, that looks a lot like a beetle I've dissected. And it turned out that this particular structure I'm pointing out is a sperm conjugate, one of those groupings of sperm. And it turned out the genus that I thought it was was in fact it once I read the caption. And so that just started, and it was around this time where I really started to just get really fascinated with these structures and want to see a point at which ground beetles have enough foundational data and understanding that we can do some really interesting evolutionary studies. And we're getting closer. And there's been some incredible work to date. So some of them have used light microscopy and SEMs to document sperm morphological variation. Here's an example of the sperm conjugate that I mentioned earlier. On the left, we've got an example where it's been viewed with differential interference contrast, and they've used a DNA stain to light up the heads where the nucleus is contained. And the little black arrows here in this image indicate this non-sperm structure that sperm are embedded in. And that's called the spermatostyle. And if we look at the bottom on these images, you can actually see that the sperm are physically stuck by their heads into this structure, and they're stuck together, traveling as a group. If I were to take a cross-section of that and look at it with a very high-powered microscope and do some special staining, I could actually see some incredible variation at the structure at the level of an organelle. And when you do that, you can see that, lo and behold, here's the spermatostyle illustrated in yellow. And you can see some examples where sperm are embedded in little cavities, little lacunae, and all sorts of details regarding, the flagellum and the axoneme and different organelles, and folks have been doing that for a while. Here's another example of a cross-section through a beetle sperm conjugate. We can see some really interesting detail to the level where there's individual cells that are very regular. So lots of very cool variation here. Those are examples of published works. And I needed to know how many species had been studied. So, and how it was distributed across the evolutionary history of the group. And so here's a phylogeny for ground beetles. And I've colored branches red that have been sampled previously for sperm morphology. And you can see that there's a good number of red branches lighting up that, folks have been studying sperm for a while now. And we've found some things out, but you can see there's actually quite a bit of white as well. Here are some arrows to indicate where those species are. And I made a point of really wanting to document to the best of my ability variation across this group, 40,000 species. It dates to about 300 million years ago. So I really wanted to do a good job sampling. And up till now, my count is 81 species out of about 40,000 have been studied. The problem is that that's very imbalanced. It actually mostly comes from two genera, Carabus and Pterosticus, which are really widely separated on the evolutionary tree. And then there's lots of gaps in between. So this is what the phylogeny looks like following David and my work on this topic. Which you can see there's a lot more red. We did a pretty good job trying to sample as broadly and densely as we could. And here's some comparisons of what the scene was like before our study and then afterwards. You can see species, there's so many species, we're still not having 1%. But getting better in terms of sampling of genera and sampling of tribes. Beetles even coming from, and to show you all, here's a map of all the locations where I got beetle specimens for this study. I studied 177 species, and that was based on 396 specimens, and here's where they come from. So you can see it's largely biased towards North America. Acquired like beetles, it was difficult to get them from elsewhere. I was able to get some funding, thankfully, and go to Southern Africa, where I was able to collect some. And this is a typical habitat for a lot of ground beetles. They love, the ones that I'm interested in particular, love sandy shores. I'm sorry I was down there. Oftentimes you'll find me flipping rocks. Here's Kojin and me flipping rocks, looking for these guys. Another great technique is setting up some bright lights and just having them come to you. And then at this point, I have to dissect them. And this is always a little tricky, especially when you're in the field, because oftentimes it means you're staying up late collecting by a light, and then you also have to stay up late dissecting. So it can be a little exhausting after a while. But what I ended up finding is that ground beetles have spectacular sperm. So here's a bunch of examples of ground beetle sperm. And I'm going to go through one example just to kind of show you some of this cool morphological variation. Again, I'm focusing on these groups of sperm called conjugates. They're composed of two parts, a spermatostyle and sperm. And here we can see this conjugate has got seemingly a good number of sperm all bunched together. But for some reason, they're only on part of that spermatostyle, and the other part is bare. It's a little odd. If you dive in and look at how the sperm heads are arranged, and this is using the DNA stain and then fluorescence microscopy, you can see that in this particular case, it looks as though the sperm heads nest one within the other. And you'll notice that there's actually some asymmetry going on these sperm heads. They actually only have the flagellums attached on one side, and yet when they come together, it appears as though flagellum is always lateral. So there's clearly some, precise construction happening within the male's reproductive tract to make all of this work out. And of course, these groups travel together in the FRT. What happens when they do? So here's an example of male sperm conjugates from male specimens, dissected from male specimens. And then here are examples of those same conjugates from those same species, or sorry, not those same conjugates, but conjugates from those same species in females, dissected from their sperm storage organ. And this is kind of crude in a way, but this is the level of data that I was able to get for parts of this project. And you can see hopefully that those spermatostyles many are bare, and they often are compressed and seemingly not as broad as they used to be. So it seems like perhaps they're being digested. Maybe there's an active secretion involved. It's not super clear. But it's a really nice example of what's called post-ejaculatory modifications to sperm. That is, changes to the sperm that happen after sperm are transferred to the female. There's also some evidence that females are really cluing into different parts of the sperm conjugate based on my study. So one was the discovery that females actually have different storage organs for different parts of the conjugate. So here on the left, I've got an illustration of a ground beetle female reproductive tract. The SPs refer to spermathecae, or sperm storage organs. And when I dissected this particular species, one of those organs contained just spermatostyles, and one of those contained just sperm. And they're separated by different ducts and came together, which is kind of fascinating. So again, kind of preliminary evidence, but it suggests that there's some morphological integration between sperm and female reproductive tracts happening in ground beetles. But as I mentioned, the focus of the study was documenting morphological diversity across this group. One of the traits that's been studied for the longest time is sperm length, and we have lots of information about variation in that. It helps maybe to have some comparison here. So ground beetles vary from about 50 microns to 3,400. Definitely more variable than mammals. Flies are wild, and then birds, a little more interesting than birds. There's also variation found in head form that I quantified for all these species. You can see some examples of that, both in terms of length and in terms of width. Here's an example of different sperm heads in different ground beetles, where there are actually species-specific differences in the sperm head, which is unusual and not seen across many ground beetles. The spermatostyle has tons of variation. And there's also a lot of variation in how sperm are placed. So here's an example where sperm are sort of distributed throughout. Here's one where they're missing from the middle and are only found on the sides. Here's an example of a species that it makes a spermatostyle with a cavity and we can restore it. Very strange. And of course, the number of sperm in the group also varies. And so these were the traits I measured. I gathered data on several continuous traits with the plan of being able to reconstruct their evolutionary history in order to study pattern and process in the evolution of sexual traits. Before I can dive into some of the analyses, I need to cover some additional examples of variation. I should mention, not all ground beetles actually have sperm conjugation. Many have just single sperm, which is interesting, and we wanted to know whether that's the ancestral state for all ground beetles or whether it's been perhaps lost. There's also examples of beetles with spermatostyles bound to a single sperm, which seemed unusual and not something that we thought would happen if the spermatostyle is just about linking together sperm, which maybe it's not. And then variation in how sperm are attached. If I take all these data and do a principal components analysis to attempt to identify the axes that show the most variance in sperm, this is what that looks like. And so we've got the first PC axis explaining about 50% of the variance and the second about 21%. You can see ground beetles, all the little dots, are actually spread fairly well throughout this morphospace, but there are some interesting gaps. There's not a whole lot of beetles in the bottom left. If we look at the loadings in order to kind of make sense of all this stuff, what traits are contributing the most to these axes, we can see that number of sperm in a conjugate, rod length, and this rod bear anteriorly are contributing a lot. And those are traits at the level of the sperm conjugate, not the sperm. And so one of the key observations in this study was that the sperm conjugate seems to show more variation than sperm and maybe evolves according to a different process. Okay, we hit a major impediment though, which is that there's no available robust phylogeny of gravity. So we did a supertree approach, and that allowed us to finally frame our results. And so here's an example of this low resolution phylogeny. It has some issues. It has branches that were not sampled for molecular phylogenetics, and it has some ambiguous placements as well. Now, what it did allow, though, was for us to begin to visualize the evolutionary history of these traits. And so here's a great one for sperm conjugation, showing that, in fact, many species have singleton sperm, and perhaps it's evolved independently more than once. If we look at spermatostyle length and sperm length, we can see that sperm length has changed a lot over time, and not always in one direction. There's a lot of convergence, it appears. And that spermatostyles indicated here in white also seem to show quite a bit of variation. And that suggests that they're evolving rapidly. Now, there's another issue with this tree. It also lacks branch length information. And that's really relevant because there's the general prediction that the distance between two species says something about how different they are, or that we can make a prediction about that. And we might think that if we had one scenario where two species diverged really recently, like on the left, versus a scenario where two species have diverged a long time ago, we might expect that there's going to be more differences between those two species on the right than on the left. And so if we were to visualize this pattern, this process, using something like simple Brownian motion, we might expect that as time goes on, that there's more differences between sampled species at the tips of a phylogeny. And I attempted to account for this by exploring different branch length schemes. I'm not going to go through all of them. I don't think it's worth it, but I feel like it's worth highlighting at least a couple. So we're taking our original tree and we're modifying the branch lengths. Here's one approach where we use Graffin's calculation. It has some good things in that all the species are the same distance from the root, but they're kind of arbitrary. It's just sort of a formula. We might also, sorry, I jumped ahead. We might also use evolutionary models. where we draw speciation and extinction rates and attempt to create more biologically realistic branch lengths, which is what I did as well. And between these two things, by randomly resolving the phylogeny in a bunch of different ways and exploring different branch length schemes, I'm attempting to explore different phylogenetic hypotheses and seeing how stable, how dependent are they on a tree versus how true are they regardless of what the tree looks like, regardless of the branch lengths. And so I now pair those data, the tree, with my sperm data, the beetle sperm data. And what that looks like in some cases is what I'm showing here, which is called the phylomorphospace, where I've taken those morphospace data and I've plotted in a tree. And I'm showing the first two PCA axes colored by conjugation state. And so one of the things that this shows is, here's some different examples of sperm heads that we're looking at in particular, that there are some examples of really small heads, really long ones, broad ones, and so on. Here's some arrows to help visualize that. But one of the things that stood out was that it appears that conjugation state might co-vary with sperm head size, which maybe isn't surprising given that the group has to come together and is stuck inside a spermatostyle. And so we see that those green dots all seem to be stuck in one spot. And it seems like a lot of the singleton sperm are kind of wandering out. They're on the edges of morphospace. But in order to address that better, we needed to know what's the evolutionary history of conjugation. This was one of our key research questions. And here's an ancestral state reconstruction for sperm conjugation. And what you're seeing is an exact character history for how this trait might have evolved. And so we see here at the base of the tree blue, towards the tips, sometimes that blue changes to other colors, sometimes it changes to orange, sometimes it changes to red. And that's one example of a reconstruction. But ideally, we want many to try and understand what was the ancestral state at the root of ground beetles. That's going to be important to know. And I played around with different branch length schemes and some different trees and found that in general, sperm conjugation with a spermatostyle seems like it was the likely ancestral state. I explored different schemes. The results tended to hold up. I explored this a little bit further by biasing it towards a hypothesis where singleton sperm is present early on in the tree. There's a group of ground beetles called nebrines that have singleton sperm. And I wanted to know if I push them so that they're sister to everybody else, what's the likelihood that sperm conjugation with the spermatostyle isn't the ancestral state? And in general, doesn't have a big effect. There's a couple of examples here, in particular branch schemes, where it maybe is a little bit more ambiguous. Each of these little pie charts is indicating the percentage of trees that had that state at that node. And so we can see in some cases, there's a little bit of uncertainty. But the spermatostyle, based on these results, the spermatostyle conjugation likely arose early on, and that ground beetles have been evolving with them for a very long time. So do sperm differ from conjugates? Seemingly yes. And I followed this up more by looking at cases where I've sampled more than one species per genus. And in general, there's a lot of variation at the conjugate level. And I'm going to move through these a little bit quickly, just because I'm trying to mind the time. But I do want to make the point that species tend to vary more at the conjugate level than the level of sperm. And so one of the things that we did is we took all of our traits and we compared these instances where we had two species sampled in a genus, and we just subtracted one trait value, one species from the other. So sperm length in species 1 minus sperm length in species 2. And so if these species are really different from one another, you'd expect those to be further away from that red line. The closer you are to this red line means those traits between these species are more similar. And the observations on the right of this plot are ones from the conjugate. you can see conjugate traits in general show more variation among species. And the point of that is that maybe there's some aspect of the underlying process that's different between these two structures. I wanted to know whether sperm conjugation co-varies with sperm form. And I wanted to ask the question, is head length adaptive for conjugation? You might think this to be the case because maybe if a sperm head gets too large, it's no longer able to fit inside that spermatostyle, or it messes with the group size or something like that. And this is in part motivated by the observation that seemingly sperm conjugates are restricted in morphospace. And so here I'm comparing sperm head length when it's singleton versus sperm head length when it's conjugated. And you can see on that density map that they do seem to be pretty different. So one way to investigate this is to attempt to apply macroevolutionary models to these data and see whether a simple random evolution model explains the data better than one where there's adaptation. And I thought it might be helpful to go through these models because they're a little, abstract without some visuals. So the classic workhorse here is this Brownian motion model that's described by a parameter, this sigma squared parameter that explains the instantaneous rate of change. And so here we've got two species, they've split off, and we can see we've got Brownian motion on this tree, bouncing this value around. And it's sort of the simplest scenario we might imagine. We might also have a process where there are actual trait optima and that there's selection, stationary selection here in our data set, pulling these species to particular trait optima. So as they're changing through time, they vary, but there's some pull towards a particular adaptive state, you might imagine. So these are great. They've allowed a lot of interesting evolutionary studies, and I wanted to apply them to our data to detect whether there's maybe different regimes in sperm head length. So I compared models. I've got one where there's just Brownian motion, and it's across the entire tree. One where maybe the rate of change is specific for singleton sperm versus conjugated sperm. Maybe they're different in some way. Or maybe there are optima, but maybe there's actually just one optimum, or maybe there are different optima. So that's what I compared, and I fit these macroevolutionary models to my data, and this is what resulted. So in each of these columns, I'm showing a different model, and on the y-axis is the relative fit of that model. And the closer you are to 0, the better the model. And one of the things that stands out is that models that include more than one regime, that say there's something different about this part of the tree compared with this part of the tree, tend to do better. Now, I just don't really have support to say that an OU process, that adaptation is happening, or that selection is operating on these traits. And instead, Brownian motion fits the data pretty well. So do sperm differ from conjugates? Yes, it seems like they do. Just to wrap up this study on developing sperm from 177 ground beetles and reconstructing their diversification to begin to generate hypotheses and also to really set the foundation for future studies focused on sperm-female reproductive tract co-diversification. So that's the next part of my talk. And this is really key because I spent a lot of time talking about male traits and leaving out this key aspect, which is female preference. And female preference is really hard to quantify. Some of you might be wondering, yes, I've experienced that myself. And part of that is because when it relates to cognition, it's very difficult to quantify. But sperm compete in the environment of the female reproductive tract. And by measuring aspects of these physical structures, we can perhaps incorporate female preference data. And that's been done. The problem is that we still have a lot of information on the univariate case. We haven't really done much work with multivariate data. And so that's kind of the scene right now at the start of my PhD. And I wanted to pursue this and incorporate female preference data and sperm data together. The goals for this study were to do exactly that, combine female reproductive tract data with sperm. I ask whether sexual selection is shaping sperm FRT traits. And then I want to know whether sperm are correlated with female reproductive tracts. There's been several comparative studies showing that sperm phenotype co-varies with the female reproductive tract, but they've generally only focused on one axis of variation, sperm length and the length of the reproductive tract, not so much all the other ways that sperm vary and all the other ways that FRTs vary. So I decided to focus on a group within ground beetles, and in particular, the Clivinini and Dishiriini tribes really stood out. And so here's an example of a Dishiriine. Here's an example of a Clivinine. There's about 1,200 of these combined, worldwide, 1,200 species. And they have some interesting traits that stood out to me. So in particular, I think morphologically, they're really exciting. So here's some examples of Clivinines and Dishiriines. Their female reproductive tracts are particularly interesting. So here I've color-coded homologous structures. And you can see there's some weird stuff going on. It's important to keep in mind that beetles are storing sperm for later use, and that, you know, how females are using sperm and manipulating it is still very much not an unknown, but there's still a lot of questions that remain. And of course, they have very unusual sperm. Sometimes they make these odd sperm with that cavity I mentioned. Some of them make these very odd broad-headed sperm. And so I wanted to know, is sperm phenotype co-evolving with FRT design? Previous research suggests that it does, and so I'm anticipating to find that these traits are correlated. Perhaps the female reproductive tract sets bounds on the dimensions of a sperm conjugate. You might imagine that if a duct is only this large, but the conjugate is this large, that it wouldn't be able to pass. So perhaps there's some simple restrictions on conjugate size. And then finally, an interesting hypothesis that maybe I won't be able to get into too much is whether the spermatostyle is maybe more than just about linking together sperm. There's been some suggestion that maybe it's about delivering important male-derived substances to the female in a timely manner. So that's also something to consider, in particular for those cases where we see conjugates that don't seem to maximize the sperm that they distribute along the spermatostyle, that instead there's big bare regions. Maybe that's part of it. So I decided to focus on these Clivinines and Dishiriines, pull from the data I already had, and gather novel female reproductive tract morphometric data. There's our friends. And as David mentioned, this required dissecting females. It's rather different from the sperm, and instead you're focused on these sort of loose structures that are surrounded by chitin, and so that's how we're able to visualize them. And I gathered simple morphometric data. I'd love to do some 3D stuff, but right now this is fairly tractable. And so I did this for 15 species. And here are some examples of female reproductive tract variation across the group. Oftentimes there is a very distinctive lobe at the end. Some of the time it's very thin. Some of the time it's shaped like this weird snail shell. There's some fantastic variation here. But again, there's a problem. And it's one you're going to hear me say before, or you've heard me say before, which is that there's no phylogeny for Clivinini and Dishiriini. And so we need that tree. And I estimated a phylogeny for the group using DNA sequence data. I gathered data from 2 nuclear genes, a ribosomal DNA gene and a nuclear protein coding gene that's been used a lot in ground beetle phylogenetics called CAD. I did sort of standard stuff, PCR and Sanger sequencing, standard molecular phylogenetics, doing the alignment, and then doing model and partition selection using a combined approach where the tree search also happens using IQ tree, which is great and we love it. The end goal is to combine these data, these sperm FRT data, with a phylogeny. And so now I've got that. But the issue is I don't know how old these beetles are. I don't know they're 5 years old, 10 years old. This is a bunch for some of the analyses I want to do. In order to get around that, I estimated divergence times using a single fossil calibration. There's a described putative Deschirrine from Baltic amber, and so I used sort of now standard approaches for node-based dating. And what that is basically is that you're saying, well, I've got a fossil, it belongs to this group in the tree, and that means that this node must be at least X old. And so that's what I attempted to do. And here's the tree that I estimated using both genes and that single fossil calibration. And so now I actually have what I need to combine these data and do comparative analyses. So one of the goals for the study was to reconstruct sperm with female reproductive tract codiversification. I wanted to know whether sexual selection is shaping sperm, and I wanted to know if sperm are correlated with female reproductive tract traits. So part of that means estimating ancestral character states, and so these are univariate estimates, and these are examples of different sperm traits. And we can focus on just a couple, and we can see that we're starting off at one point on the left, and then as evolution happens, we see a lot of diversity on the right. And in some cases, like on the left here, we can see that the tree is kind of tangled. There's a lot of species that have converged on the same trait value. Whereas on the right, we can see, it seems like there's some separation. There's one group over here and one group over here. We can do the same for the female tract traits. And again, this is necessary in order to understand how these traits are evolving. Simple characterization. We can also use a multivariate approach, which is really exciting, by plotting a tree in morphospace. And that's what I've done for sperm on the top and female reproductive tract data on the bottom. And one of the things that stands out is there's some imbalances in how Deschirrine and Clivenine have explored sperm female reproductive tract morphospace. So one of the things that stands out to me right now is that it seems like Deschirrines, there in blue, are really restricted in terms of their exploration of female reproductive tract morphospace. So that makes me think that likely that's led to a corresponding restriction in their exploration of sperm morphospace. And we can see they've explored much more along one axis than another. Clivenines, on the other hand, in red, seem to have diversified a lot in female reproductive tract morphospace, and also a fair deal in sperm conjugate morphospace. So one of the questions I asked was, are these traits evolving with selection? And so similar to my previous study, I'm going to be modeling evolution, comparing the fit of a Brownian motion model with an Ornstein-Uhlenbeck OU model. And looking at tables is boring, but one of the things you can tell is that some of the time Brownian motion really stands out as being a good fit. And some of the time OU really stands out as being a good fit. And interestingly, many of the sperm conjugate traits are better supported by an OU model, which means there's selection, there's a pool, there's an optimum, which is exciting if that's the case. But I only have 15 species. And so in order to confirm this result, I did some simulations where I made up data under a known model, and I try and see whether I can actually recover the model that generated the data. And if I can, that's great. Then that means I probably can believe in my results. But if I can't, then that probably means I can't. So here's an example of some simulations. This is a simulation with Brownian motion. I've done about 1,000 of them. And on the y-axis is the relative support of the model. And so the closer towards 1 you are, the more support that model is getting. And you can see here, OU is colored in teal here. And a lot of the time, they're getting, 70% of the relative support, which is really shocking. And that means that OU wasn't used to make these data, and yet OU models fit really well. So what if you explore other parameter settings? This is important to do. When you do that, you can see a largely similar pattern. So on the left, I've generated data with Brownian motion with different rates of variation, seemingly same results between runs. And then on the right, I've generated data with OU and seen if I can recover an OU model. And one of the things that you can see there a little bit is that as the strength of selection increases, which is what I've done from one plot to the next, an OU model becomes clearer. Once the strength of selection is super strong, it really is quite clear that a Brownian motion model doesn't fit well. Okay, so what this suggests is that there's a power issue. I probably can't say that my traits evolved via OU, or I can't reject Brownian motion, maybe would be a better way to say it. I was interested in estimating whether there's covariance between sperm and female reproductive tracts. And I used a couple different approaches. One is phylogenetic generalized least squares, which is very similar to sort of standard regression, except it's phylogenetically informed. And when I did that, I found that actually, in fact, some of the sperm traits are indeed correlated with some of the female reproductive tract traits. And they tend to be the traits that are involved in sperm conjugation and not so much sperm themselves. I also repeated these analyses using multivariate modeling. So we might have a situation where two traits are evolving, but there's no covariance between the traits. That might be one model. There might be a dependent model of evolution where there's some rate explaining how quickly sperm evolved, there's some rate explaining how quickly female reproductive tracts evolve, and then there's this covariance element. And you can compare these two models in addition to doing regression. Regression is sort of tried and true. This is another approach for looking at covariation in a tree. So when I do that, I find out that actually a lot of these traits are correlated through time, that many sperm conjugate traits are correlated with the female reproductive tract. The length of the spermatostyle is positively correlated with the female reproductive tract length. The spermatostyle width is correlated with the FRT width, and the number of sperm in a conjugate as well. So that was kind of interesting. I was excited to see that. So what did I learn from this study? Well, I learned that sperm and female reproductive tracts have dynamic evolutionary histories, that there's evidence for morphological integration or correlation between sperm and female reproductive tracts, and in particular, sperm conjugate traits appear to be correlated with several dimensions of the female reproductive tract. And really, although I can't say this definitively, the results are consistent with the scenario where the female reproductive tract is exerting stronger selection on sperm conjugates than sperm, which might be indicative that the spermatostyle is maybe more than just about linking together sperm. So with that, I'm running out of time here, basically, but hopefully you all can stay a little bit and hear me just thank the amazing people in my life who have made this work possible. My lab mates have been incredible, and 7 years feels like it went by really fast. I want to thank my funding, National Geographic, the NSF. I also need to thank the Entomological Society of America for funding part of my field work. I also really need to thank my committee. That's misspelled. You all have been amazing and really pushed me to believe in myself, even in moments when I definitely did not believe in myself. And I just really know I couldn't have done it without you. I want to thank the IB staff and grads for all their help in terms of making OSU such a welcoming environment. The community of ground beetle workers who make things fun and share data and keep me excited for the future. And then you all who are studying sperm and making incredible discoveries. Because as you saw in that timeline, there was a lot of white men, and now we're in a zone where there's a lot more contributions from a lot of different people, and it's wonderful. And so I feel like that's worth celebrating. I also wanted to very quickly end by thanking some of my field companions. James in particular here on the left was crucial to some of our trips. For instance, he fixed that flat that I got, and then, well, he also fixed the flat a second time, and he also fixed a flat a third time. So I really appreciate the friends I've had along the way on my collecting trips. I really want to thank really just the people who have made such a positive impact on my life. And I think with that, I will end and take any questions you all have.
Thank you, Antonio. And I'll clap for the whole group.
Thank you, David. I'm seeing some little Zoom claps.
Good. Excellent. Excellent. So now we'll have questions from the audience as a whole. And you can feel free to either ask it in the chat or just unmute yourself. And this will be non-committee members. And then after a little round of questions from the audience, then we'll retreat to the part of the meeting in which it's just the committee and Antonio. Any questions?
I'm just enjoying the the chat log. Thank you. I really appreciate it.
Questions?
Can I ask a question?
Yes.
Sure.
Dave.
Good. Antonio, I may have missed it, but what kind of outgroup did you look at to find out about what the features were at the base of the Carabid tree?
That's an awesome
What's the
question.
status of conjugation outside of Carabids, or is there any?
Yeah, so Dave is asking about the distribution of sperm conjugation maybe throughout beetles or throughout the group that includes ground beetles. And it's really interesting. So adephagans have just very bizarre sperm. In terms of to answer your question, in terms of the outgroup sampling that I included for that particular reconstruction, it actually only includes ground beetles. And so, as you probably are imagining, a much better way to do that would be to include some outgroups. Seems like maybe gyrinids are sister to ground beetles, and the earliest diverging gyrinids have singleton sperm, but some of the ones that are nested deeper within the tree have sperm conjugation that looks very similar to what ground beetles have, which maybe means that it's evolved convergently in these two groups. That'd be incredible. If you expand beyond and look at haliplids and friends, there's just a lot of really weird conjugations. The diving beetles are great examples. So yeah, no, there's a lot of fun examples there. Weird sperm.
John.
Okay.
I see your hand up.
Hi, Tony. That was really great.
Thanks, John.
You sort of tickled on this point about the spermostyle at a number of points. I was wondering if there's any work that might identify what the spermostyle is made of, and if it might point to the spermostyle being perhaps a nuptial gift, particularly because it's sequestered by the females, separately. Maybe that's a driver in the evolution of these conjugates. Maybe if there's something the male has to offer to protect the female, the young, maybe that's why these spermostyles, the conjugates evolved. I didn't know if you had anything to comment on that.
No, I can just like blow a bunch of hot air.
Oh, okay.
But it is something
Do
that we are really interested in, and that isn't to say we totally don't know. There have been some staining studies, some histological studies, and it's clear it's got a lot of carbohydrates and a lot of proteins. Other than that, you know, it's really hard to say. But the fact that some species have evolved seemingly separate storage organs for different parts of a male sperm conjugate in these beetles, and the fact that most ground beetles have it seems interesting. And a lot of people have looked at the evolution of sexual traits, but not many people have considered when those traits are lost. And so if sexual selection is maintaining sperm conjugation, it's kind of interesting to think about what's happening in those lineages where it's lost. Is it that natural selection is now overwhelming sexual selection? Did sexual selection relax? Those are the sort of things that you can do once you've got a good tree. But no, to answer your question, nothing is known for sure whether it's a nuptial gift, but it could be. That's one of the hypotheses for sperm conjugation.
Seems like a potentially fertile area. Pun intended.
Nice one, John. Nice one, John.
Okay, so let's, one more question if there's one out there. And then we should move on. Okay, well, if not, then let's thank Antonio again for an excellent job.
Thank you, everybody. I really appreciate your coming. Oh, whoa, we got Javi, we got Carolina. This is amazing.
Felicidades, Toño.
Oh, gracias, Caro. Ciao.
So,
Oh, man, I wish I was in Coevalence with y'all. And thanks for the Syracuse people in France. This is amazing.
so for the committee, let's actually, why don't I'll quickly send everybody an email with a different meeting link. Or we can just wait till everybody signs out. Let's just wait till everybody signs out. See you,
See you,
Scott.
Scott.
See you, Jean. See you, Laura.
Oh, wow.
Let's see. Who do we
I
have
like
left
Devlin's
here?
image.
Why?
Got my aunt Mirna. Hi, Mirna.
Okay. Teresa,
Yeah,
are you there?
you ready? I'm
Yep,
here.
we're done.
Okay, cool. I'm going to end it